to the next talk, which will be given by Ekaterina Paramonova from the Institute of Mathematical Problems of Biology, Keldysh Institute of Applied Mathematics, Russian Academy of Science. And the name of the talk will be Grid Density Functional Study of Iron Impurities in Hydroxyapatite.
Hello. Hello.
Oh.
Yeah. Okay. Hello, everybody. The topic is density functional study of iron substitution in hydroxyapatite. Okay. What is hydroxyapatite? It's a biomineral component of bones of vertebrates, main component. Its formula is given here. It's calcium 10 PO, then for PO4 6 and hydroxyl group. But we have two different positions for calcium. It's calcium 1. It forms calcium channels. In the structure, and calcium 2, it forms those walls of channels with hydroxyl group, calcium columns in the structure. The structure of hydroxyapatite, it can have a lot of impurities. For several positions, there are a list of substitutions, and of course, it influences on the properties of the material. Why we choose iron-doped hydroxyapatite? Because of its high biocompatibility, it could be used in medicine. For example, as magnetically induced heat determination of cancer and so on. On the slide, there is a structure of pure hydroxyapatite. There is it's a supercell on 2 by 2 by 2 unit cells of hydroxyapatite. It's here. Here there are those calcium columns. Just a moment. Here. Oops. Sorry. Okay. It's not seeing the mouse. Yes. Okay. We consider different types of impurities here. It's substitution of calcium 1, calcium 2, and phosphorus onto the iron. And here with D, B, C, we made an interstitials. In previous work, there were done tests on methods. We show that the use of hybrid density functional theory with HCE will be good. In the current work, we use HCE functional realized in quantum espresso. But again, this supercell is quite big. It contains more than 300 atoms. It's 352. So we use for defective structure, we made geometry full relaxation with PBE functional, and then we made a single point calculation with hybrids HCM. So then here there is a formula for formation energy of a defect in a crystalline sample. It depends on the energy difference between defective, it's EQR, and pristine crystal. And the third and fourth terms, they account for any stoichiometric and charge imbalances. Here, mu eta I are the chemical potentials for any species which are added or removed from the perfect crystal to make it the defect. Those chemical potentials have a limited range, can vary in limited range here. So we came to those diagram huge from where we should consider focus on the following extreme cases: calcium rich and phosphorus rich, then calcium poor and phosphorus poor. It's about synthesis. Okay, here are the neutral iron defects. It's after in small parts within 4 angstroms neighbors of ferrum in after optimization. No. In some cases it's D. For example, ferrum causes the flipping of one of hydroxyl groups. And also we calculated charge iron defects, and here with that hydroxyl group, after optimization, after adding ferrum, iron atom comes to the wall of the channel, and after we try to make it backward reaction, it's not going on. It still has the same geometry. Also, that was done for interstitials, and we came to formation energy diagram with different defects, charge one and neutral one. And we have different conditions for calcium poor conditions. It's A. The charged substitution of calcium 1 will be preferable than charge substitution of calcium second. And while at the neutral cases, it will be otherwise. It's okay. Okay. Sorry. Conclusions of there was a discussion in this and previous papers about what are the more probable substitution defect. Is it a substitution or interstitial? We came to both interstitials and substitutions irons are allowed in iron substituted iron defective hub, ferrum hub. It depends on conditions of preparations also. In the calcium poor conditions, ferrum calcium substitutions are preferable. Okay. In the calcium rich conditions, the iron interstitials in the hydroxyl channel are preferred. The neutral and single charge defects with iron are placed precisely between two hydroxyl groups. At higher charges, the structure is not stable and transformed to the one where iron is displaced as the channel wall. The opposite process of discharging does not return the high symmetry structure. Iron remains attached to the oxygen, so the channel wall. At phosphorus poor conditions, we predict the overwhelming iron for phosphorus substitutions, probably in the form of replacing of entire phosphate group by ferrum oxygen 1. But we haven't that in the literature, considering the literature. Okay, and highest magnetization was calculated for charge substitution ferrum plus calcium 1 and ferrum free interstitial. Okay, thank you.
Thank you. So maybe we have some questions. If such, we have a time to ask.
Yes, I have question. Possible?
Please.
Okay.
Yes. Sorry, Ekaterina, I not full understand. So maybe it is the most preferable is ferrum substitution in this what to say first case when you have calcium poor. So it is neutral position, but ferrum is plus. Can you more detail explain what is most preferable in this case? It's very interesting. So
in calcium poor?
Yes. Which is ferrum substitution in this case?
If neutral, it will be calcium 2. Substitution for calcium 2 on ferrum.
In calcium 2. So it is some.
It's a wall around hydroxyl group.
Okay. Yes. And it is maybe still some along OH channel between two OH hydroxyl or not?
No. It's substitution of calcium that forming that channel.
No, close to the channel.
That one, yeah, like okay. Here, right.
Okay. So you calculate this by WASP or by quantum espresso?
Quantum espresso.
Okay. So it's such recent data. Okay. Thank you very much. Good. Okay.
Thank you.
Thank you very much. Maybe more questions? Okay, we don't have questions. Okay. So unfortunately, the last talk in the session, the speaker said that he cannot take a part in the conference now due to her own reasons. So it means that we should skip the last talk in the session, and we are about to finish. So I want to acknowledge everyone speaker in this session, and we will meet after lunch, I guess, and see you later. Bye-bye.